Okay, welcome to Ask the Expert. Oh, here he is. Today we are speaking with Dr. Abhishek Jain. He's the Associate Professor, Department of Biomedical Engineering, College of Engineering at Texas A&M University. And also Tanmay Mathur, who is working with him in this project. Together, they are working in the space of organs on a chip. And I'm going to let them do a little bit of introduction on what it is they're pursuing at Texas A&M. So welcome and thank you for joining us. And yeah, take it away. If you have slides, you can start now.
Thank you for the introduction. Start sharing my screen.
So Tommy, are you a graduate student or postdoc?
So I'm a senior grad student. I'm about to defend. So
oh, great.
Yep. All right. Good morning, everyone. As explained, I'll be talking about engineering diabetes and personalized and vascularized organ chips. Specifically, we will be talking about blood endothelial progenitor cells on a chip, a disease and patient-specific cell source for in vitro vascular tissue engineering platforms. So, a major challenge in modern medicine is its lack of specificity. Different individuals respond differently to the same treatment. In fact, studies have shown that only 4 to 25% patients show positive signs of recovery. Then there's these large group of non-responders. And finally, we have up to 20% patients who show negative adverse side reactions. This can be primarily attributed to the fact that we do not have physiologically relevant humanized disease screening models. On the other hand, personalized treatment can significantly improve patient health, and it can significantly reduce costs. But as I said, we currently do not have tools which can allow us to assess personalized treatment. This is where the organ-on-chip technology comes in. These microfluidic platforms can be functionalized independently and cooperatively to mimic physiological tissues or physiological flow patterns. For example, in the context of vessel chips or vessel organ chips, we can functionalize these devices with human-specific cells like endothelial cells, humanized endothelial cells. We can perfuse whole human blood through these devices, and finally, we can manipulate the flow patterns through these devices by connecting them to programmable pumps. So these platforms can allow us to perform a dissectable investigation of the contribution of each of these factors to the pathogenesis of any disease. However, that being said, even the current organ-on-chips or vessel chips primarily have certain limitations. Most of these platforms utilize cells that have been isolated from healthy individuals. The figure in front of you shows some common primary endothelial cells that are being used in the vessel chip platforms. And as I said, these are isolated from healthy individuals. So it is very difficult to model disease with high specificity using these primary cells, and we cannot perform a patient-specific investigation using these cells anyways. As an alternative, with the advancement in iPSC differentiation technology, researchers have been able to differentiate induced pluripotent stem cells into mature endothelial cells. However, this process is also time-consuming. It requires sophisticated training and requires expensive reagents, which might not be accessible to a lot of groups. This is a common differentiation protocol that has been published to obtain endothelial cells. As you can see, different growth factors need to be administered at different times, and a small error in the time of administration can cause catastrophic changes in the final yield of your product. So the need of the hour is to find a suitable alternative endothelial cell source, which is relatively easier to obtain and can differentiate and is disease specific and patient specific. So we have identified one such cell source, which we call blood outgrowth endothelial cells. These studies have shown these are late stage endothelial progenitor cells found in circulating blood. Studies have shown that BOECs are increased in cardiovascular patients. They are highly proliferative. Once we isolate them from blood, we can obtain more than 1 million cells within 2 to 3 weeks of expansion from a very small number of cells present initially in blood. Once isolated, they exhibit the classic endothelial cobblestone morphology, which is similar to all other endothelial primary cells that we have been using. And compared to iPSC endothelial cells, it's easier. BOEC isolation and expansion is relatively easier and straightforward. That being said, really the hypothesis that we are trying to test here is that are these BOECs that are being derived from diseased patients, are they able to carry the disease phenotype? And can they mimic this in vivo dysfunction that we are observing on any in vitro platform? So this is a small video showing a BOEC lumen on one of our microfluidic chips, which was 200 micron wide and 75 micron high. So this video shows that, yes, we are able to culture these cells, and these cells are able to make 3D confluent lumens. But before we could go forward and test our hypothesis, we wanted to characterize these cells compared to existing endothelial cells like HUVEX and iPSC-derived endothelial cells. So we, off the top of the head, we just compare the morphology of these cells, and as you can see, they exhibit the classic cobblestone morphology, which is also observed in primary human umbilical vein endothelial cells, as well as the iPSC derived endothelial cells. Then we also wanted to study the surface marker expression of BOECs for common endothelial markers like weak, adherent, CD31, and VEGFR2. And as expected, the expression levels of these three markers in BOECs are similar across compared to these other two endothelial cell types. We also wanted to study how these cells behave in terms of their growth kinetics, and if we can monitor that. So we performed a scratch assay with these three cell types, and as expected, there is no statistical difference in the growth rates between the three cell types for the first 24 hours, showing that BOECs exhibit comparable growth rates and migration capabilities in vitro compared to the two other endothelial cell types. We then also performed RNA sequencing because we wanted to study how BOECs differ to HUVEX or iPSCs in terms of their transcriptomic profiles. So we performed RNA sequencing, and we find that there were around 14,000 genes which were commonly expressed between the three cell types. However, when we compared BOECs and iPSCs independently to HUVEX, we find that BOECs were more transcriptomically closer to HUVEX compared to iPSCs, as you can see from these correlation plots at the bottom. On the right-hand side, we can also see the Pearson correlation coefficients. A higher Pearson correlation coefficient means that BOECs are closer to HUVEX in terms of their expression profile. And hence, these results suggested that BOECs might be functioning similar to HUVEX, transcriptomically at least, compared to iPSCs. We then further performed differential gene expression testing in iPSC, ECs, and BOECs compared to HUVEX. And what we find interestingly is that iPSC-derived endothelial cells had more differentially expressed genes compared to HUVEX. On the other hand, BOECs had lesser differentially expressed genes compared to HUVEX, which suggests that BOECs are actually behaving like HUVEX because they have lesser number of genes which are differentially expressed compared to HUVEX. So the transcriptomic results so far showed that BOECs compared to iPSCs were transcriptomically closer to HUVEX. They were behaving like HUVEX transcriptomically at least. We also then finally wanted to study how these cells respond to shear. As everyone knows, endothelial cells inside the body, they're always facing or they're always witnessing fluid shear stress, and that is what they like. So we wanted to study how each of these cells is responding to fluid shear stress. So we cultured these three independent cell types in a straight microfluidic channel, which was 200 micron wide and 75 micron high. You can see the image on the left-hand side. We cultured these cells independently under constant media perfusion, which were at 1 microliter per minute, which for this particular design resulted in a shear rate of around 100 second inverse, which is a typical arteriolar shear rate found in a blood vessel of this size. So as expected, when we performed a time-lapse study, we find that the BOECs exhibit a similar growth response under shear as well. And interestingly, what we also observed was that BOECs did not differ in terms of their orientation with respect to the flow direction, their average cell area, as well as their average circularity. Both without perfusion and with perfusion uh, compared to all other cell types. So overall, BOECs exhibited comparable growth rates and migration capabilities in vitro under perfusion compared to other endothelial cells. So summarizing the results that we have so far, what I've shown so far is that healthy BOECs are actually similar to primary cells like UX and iPSC-derived endothelial cells. They express endothelial markers. They express the standard cobblestone endothelial morphology. They express similar lumen integrity. They also show same shear responses, and they also have similar responses to exogenous stimulation. But what about our hypothesis? What about disease specificity? So to test my hypothesis of disease specificity, we focused our attention on modeling endothelial dysfunction in type 1 diabetes. Studies have actually shown that endothelial progenitor cell counts are actually reduced in type 1 diabetic patients, and the proliferation rates of these EPCs is further reduced in diabetes. And because of the hyperglycemic conditions that exist within type 1 diabetic patients, this leads to oxidative stress, which further leads to endothelial dysfunction. So we wanted to test the hypothesis that BOECs isolated from diabetic individuals would be able to mimic these hallmarks which have been observed clinically on our in vitro platform, and that they would be much more inflamed or activated compared to our healthy BOECs. So we went ahead and we isolated BOECs from healthy controls as well as diabetic individuals. And as expected, we find that diabetic endothelium was not able to reach confluence under the same time that the control endothelium required to reach confluence.
Wow, that is really remarkable. That's such a difference morphologically.
Yeah, and culturing them further, we found that they actually took roughly twice the amount of time to reach confluence, which basically showed that yes, there is some difference in their proliferation rates. We also wanted to study their actual metabolic activity using Alamar Blue or MTT assays. So we used Alamar Blue both on a well plate format and our vessel chip format. And as expected, the diabetic endothelium had reduction in their metabolic activity with time. And this was observed both under the flow as well as static conditions. We also wanted to test the presence of reactive oxygen species in both healthy as well as diabetic BOECs. And when we performed the oxidative stress analysis, we find that diabetic endothelium is definitely, it has more oxidative stress compared to the control cells. Finally, we wanted to study how these BOECs differ in terms of their endothelial activation and can they induce thromboinflammation. So we cultured healthy control BOECs in our microfluidic channels. Then we also cultured diabetic BOECs in these microfluidic channels. And as positive controls, we also cultured primary endothelial cells. And we had another condition where we had exogenously stimulated those primary endothelial cells with TNF alpha, 5 nanograms per ml TNF alpha. And as expected, after a whole blood perfusion experiment for about 15 minutes at arteriolar shear conditions, as expected, we do not find any platelet adhesion on the untreated primary endothelial cell channel as well as our control BOECs, which shows that the control BOECs were not inflamed at all. They were behaving as regular endothelial cells in vitro. And as expected, the TNF alpha treated device had more platelet adhesion compared to the untreated device. And finally, interestingly, we find that the diabetic BOECs had the highest amount of platelet adhesion to its surface, showing that diabetic BOEC was indeed activated without any exogenous stimulation compared to any other, or compared to the control BOEC. Quantifying these results, we can see a dramatic increase in the platelet coverage compared to control BOECs. So these results collectively so far show that diabetic or diseased BOECs are able to mimic the physiological behavior that we are observing clinically on any in vitro platform, and that control BOECs behave like healthy cells. So we can say that BOECs might be suitable to study disease specificity, but so I'm not showing results of the work that we have done ahead of this. So The work that I've shown so far has been disseminated in 3 journal papers. The journal that you can see in the middle, we wanted to study, or we wanted to test our hypothesis, and we wanted to go one step further. So far we had shown that these cells are disease specific, but we wanted to test if these cells are also patient specific. Are these cells sensitive enough to differentiate different patients within the same population?
The big question.
Yeah, sorry. Go
No,
ahead.
I said that's the big question for
Yeah, that's the
personalized
big
medicine.
question.
Yeah,
Yeah,
fantastic.
so we didn't test that hypothesis with type 1 diabetes. We focused our attention on modeling endothelial dysfunction in sickle cell disease. But in my second manuscript, we have shown that when we perform the same analysis with BOECs from multiple sickle cell patients, we are able to see differences in their hallmarks, both transcriptomically and functionally on our vessel chip platform. So overall, BOECs might be they might be a suitable alternative to current endothelial cell sources, as they are relatively easy to obtain compared to iPSC-derived endothelial cells. So in summary, these vessel chips made from blood may serve in preclinical discovery and personalized assessment. And I would like to mention here that although we have focused our attention on modeling endothelial dysfunction in type 1 diabetes as well as sickle cell disease, in theory, this hypothesis can be extended to other diseases as well, like cancer-associated thrombosis, deep vein thrombosis, beta thalassemia, etc. With that, I would like to end my presentation, and I would like to thank my team, especially Dr. Jain, for guiding me throughout this journey, as well as our funding sources. We got funded. This project was funded by the 2018 Trailblazer Award, which was awarded by the NIH NIBIB. And I would also like to thank Texas A&M University and the Biomedical Engineering Department here at A&M. That's it. If I would like to answer any questions.
Thank you, Tanmay, and soon to be called Dr. Mathur.
I hope.
I'm sure. This is a really, really excellent body of work. And it's so interesting. This model system, right, could be really used in many places. You could, you could use it in the prodrome, those who have just one autoantibody to type 1 diabetes, 2 antibodies, and see how that impacts those folks, right? Or you could also look at sort of post-diagnosis implications in the vasculature and the circulation. This has a wide a reach into type 1 diabetes, as far as I can see. I'd love to hear a little bit from Dr. Jain. What are your thoughts on where this might, go? I think you have to unmute yourself.
I agree with you, and that is our hypothesis. This is something that we found almost serendipitously. These outgrowth endothelial cells have been studied for a couple of decades now, not too long ago, but for at least 10, 15, 20 years. And, there is no clear, well-defined characterization or even a definition of what these cells particularly represent. But when we isolate these cells and when we put them together in these organ chips, we see that they actually exhibit a lot of these symptoms that you actually see clinically in some of these patients. So in the context of type 1 diabetes, for example, I think at least for some of these advanced patients who might be having these downstream or collaborative stroke-like symptoms that are hard to track or hard to diagnose, and there are some of these markers are not available or not very specific within the blood cells, you cannot do biopsy of actual tissue. Maybe these outgrowth endothelial cells might be telling us a story. They might be telling us something about these patients that the clinicians need to know early on in some of these advanced patients. So
yeah, I mean, they could certainly be, kind of helping clinicians to find progression to
correct,
problems. But yeah. I also think that there could be a real utility for this technology or this, model system in tracking changes during the prodrome. As people progress to type 1, what is happening? even before they even, become diagnosed with type 1, we know that there are certain stages of progression and that those stages correspond with different biomarkers. But I mean, this might even give more insight into things.
Absolutely. And I think when you mention early stage, so I'm not 100% sure at this stage whether, vascular endothelial markers are of relevance for early stage diabetes. Maybe they are, and maybe we don't know, maybe we know about it, but we haven't looked at it. So if it turns out that they are important, then this could be a very, very valuable resource to track down early stage patients and find out some of these symptoms at an early stage as well. I completely agree. And the fact is that these are all blood resourced cells. So, it's, a patient goes to the clinic, they do a blood draw anyways. So, you could very easily add that into the biomarker panel that you would not do otherwise and not routinely done, but this could be done.
And how easy would that be, to, the scale, to scale it up to patients who are just entering their prodrome or have their first autoantibody? And
Right.
Who would you coordinate with? Would this be like through TrialNet or, how would it work?
Yeah, so this is a lab assay in the sense that it's not a bench. I don't see it as an assay that you could do it in the clinic as an instant readout. I think you would have to wait for results for maybe 2 days or 3 days, and the sample will have to be sent to a specific diagnostic lab or a company that would do the assay. They would actually isolate the cells, culture them, which would take a day or so, and then they will send the result back. So it's not an emergency care type of a diagnostic where you need an instant result.
Yeah.
But it would maybe take a day or two to get it, but it would still be faster than a biopsy result.
Oh, sure. And
Right?
what about,
So somewhere in there.
What kind of companies might be, potential targets to do this assay?
I would be excited to see if, a company like Quest Diagnostic or Abbott pick something like this up, and they say, okay, we want to invest. They already have the tools, they already have the infrastructure to do blood assays. So they have everything in place. So they could adopt this kind of a technology and put it in their mix of diagnostic as well, when diagnostic is of interest.
Yeah. Do you think they would require some kind of laboratory, verification that there actually are changes first? I think so, right?
If you do you mean like it will require like an FDA pathway to get
maybe just like, some kind of paper showing that there are, this system can detect changes as early as, the advent of the first autoantibody.
Absolutely. I think this will require some form of more advanced clinical validation.
Mm-hmm. Yeah. Are there any questions in the audience? I see there's a couple people here. I saw someone from industry. Okay. Maybe not today, but yeah, I think this is really, really interesting. And can I ask also, has anyone or your team looked at sort of genetic changes in the BOECs or the blood outgrowth endothelial cells? Have you noticed anything?
So we have done a very rigorous, RNA sequencing of these cells. So we have all of those data sets available to us that we have actually published, and we have also put them in a repository. So we have done this characterization. We have looked at a variety of pathways that get activated in patient BOECs relative to normal. So the normal BOECs are very close to, primary endothelial cells.
Yes.
But these patient cells, they exhibit a lot of these signaling pathways that are upregulated, a lot of these genes getting upregulated that you would expect to see in a patient who has vascular disease.
Yes. So you have sort of like time stamps or, kind of different, snapshots of these different genetic changes, I guess is what I'm asking.
Yeah, we have also looked at some time course changes as well.
Right.
So we have some data on that as well.
Fantastic. All right. Well, if anyone is interested in reaching out to the Jain group, And maybe, in a collaborative way, it sounds like this is, this system that you've developed is ripe for more exploration. And, congratulations to Tanmay Mathur as he defends his thesis and goes on to the next thing for his postdoc. And I would also just comment that, he's well versed in this system. So I'm sure he's going to have a lot of options. So congratulations. And I can't wait to see what your laboratory does next.